Hello, this is Dr. Stewart Tepper, Professor of Neurology at the Geisel School of Medicine and Director of the Dartmouth Headache Clinic. I'll be discussing the publication Safety and Efficacy of ALD403, an antibody to calcitonin gene-related peptide for the prevention of frequent episodic migraine, a randomized, double-blind, placebo-controlled, exploratory Phase 2 trial by David Dodick and colleagues. This study report was published in Lancet Neurology. I selected this article to discuss because episodic migraine is a common and disabling disorder that is ineffectively treated in many patients. Calcitonin gene-related peptide, or CGRP, is an important neurotransmitter in the pathogenesis of migraine, which has led to the clinical development of several CGRP antagonists, or GPANTs, and anti-CGRP monoclonal antibodies. ALD403 is a monoclonal antibody that potently and selectively binds to both alpha and beta forms of human CGRP. The goal of the present study was to assess the safety, tolerability, and efficacy of ALD403 for the prevention of episodic migraine in adults. Methods of the study were: this was a Phase 2 randomized, double-blind, placebo-controlled, multi-center proof-of-concept study involving adults with a 12-month or more history of episodic migraine. Patients were allowed to use acute migraine treatment for up to 14 days per month, up to 10 days of triptan use. Patients could not have used standard preventive treatment for more than 7 days during the 3 months prior to the study and were not allowed to have used onabotulinum toxin A for the previous 6 months. Patients were randomized 1-to-1 to a single dose of ALD403, 1,000 milligrams, or placebo via an intravenous infusion. Randomization was stratified by baseline number of migraine days per 28 days. Key findings of the study were 174 patients were randomized, of whom 163 received study treatment. Patient mean age was 39 years, 82% were women. At baseline, patients experienced 8.4 to 8.8 migraine days per 28 days. The Headache Impact Test 6 score was 63.8 to 64.5, indicating severe disability. An adverse event was experienced by 57% of ALD403 patients and 52% of placebo patients. An upper respiratory tract infection was the only adverse event that occurred in 5% or more of patients and more frequently in the ALD403 group than the placebo group. A URI occurred in 9% of ALD403 patients and 7% of placebo patients. No treatment-related serious adverse events occurred in either group. The mean reduction in migraine days between baseline and weeks 5 through 8 was 5.6 in the ALD403 group and 4.6 days in the placebo group, and the difference was statistically significant. Similar results were observed during weeks 9 through 12. Here are my thoughts and analysis of this study. This Phase 2 trial found that one single infusion of the anti-CGRP monoclonal antibody ALD403 prevented episodic migraine with good efficacy, safety, and tolerability. Because this is an experimental medication in an exploratory Phase 2 trial, no change in current patient management will occur yet. ALD403 and the other two anti-CGRP monoclonal antibodies, fremanezumab and galcanezumab, as well as the anti-CGRP receptor monoclonal antibody, erenumab, all prevented episodic migraine in Phase 2 trials. ALD403 is the only one of the four which was given intravenously in a single dose. As was noted, larger Phase 3 trials of efficacy and safety need to be completed. Should these trials confirm the Phase 2 results, there'll be an intravenous option for administering a preventive biologic in migraine prophylaxis. Several unanswered questions remain on ALD403, assuming efficacy, safety, and tolerability are confirmed. First, is there any advantage to an intravenous formulation for patients over subcutaneous injections? Second, how long will the therapeutic effect of a single infusion last? This latter question is being studied in a longer trial of ALD403 underway at the time of this recording.